Hello students, myself Popere KS, Assistant Professor in Botany at Red Shikshan Sanstha's RBNB College, Srirampur. In this lecture, we are going to study about the gene regulation in case of prokaryote. This gene regulation, it occurs at the different stages like transcription, translation and at the replication stage. So in this lecture we are going to study about the gene regulation at the transcription level. So this gene level at the transcription level, at this there are two ways by which the gene regulation will take place. That is, first is negatively regulated system and next is positively regulated system. So first, so these are the two ways by which the gene regulation will occur at the transcription level. So first is negative regulated system and next is positive regulated system. So in case of the negative regulated system, in this there will be an inhibitor which will bind to the promoter and it will inhibit the synthesis of or it will inhibit the transcription process. So in case of the negative regulated system, there is presence of an inhibitor which will be stopping the transcription process or the initiation of the transcription. Now at antagonist to this, so there is a presence of inhibitor. So antagonist to this, there will be also an presence of inducer which will allow the synthesis of or the transcription process to start, or it will initiate the process of transcription. So at antagonist to the inhibitor, there is also an presence of inducer. So in a positive regulated system, there will be a presence of an inhibitor which will stop the transcription process, and antagonist to this, there is presence of inducer which will start or initiate the process of transcription. Now next is the positive regulated system. So in this positive regulated system, there will be some protein or some enzyme which will be acting as which will allow the synthesis of mRNA or the transcription process to occur. So this effector molecule So there is presence of an effector molecule. The effector molecule, it may be a protein, small protein or a large macromolecule. This will allow the synthesis of mRNA or the transcription process to occur. Now next we are going to see the negative regulated system. It occurs by two ways. So first is inducible and next is repressible system. So first we will see the inducible system or the inducible operon. That we are going to see about the lac operon. Now this inducible operon or lac operon. So this is this lac operon, it is a setup of a gene. So there are regulatory gene and structural gene. This the structural gene, their regulation is done by the regulatory gene. So let us see the structure of this gene. So this is the lac operon, and this is the promoter. This is the lac-I gene. This is the promoter of the lac-I gene. Now next, this is the promoter and this is the operator of this 3 structural gene, that is so these are the structural gene, that is lac-Z, lac-Y and lac-A. Now the synthesis or the transcription of these 3 genes, that is lac-Z, lac-Y and lac-A, is controlled by a single promoter, or so this is called as a polycistronic. So this is a polycistronic gene in which the transcription of this 3 structural gene, it is controlled by a single promoter. So these 3 are the structural gene, and this lac-I is a regulatory gene. Now we will see how the lac operon works. This lac-I, this is an inhibitor. So this lac-I, promoter of this lac-I, it will after transcription it will produce a protein or an enzyme. This will acting as an inhibitor. Inhibitor means in the presence of this inhibitor there will be no synthesis of this structural gene, or the transcription of this structural gene is not going to take place. So this is an inhibitor. This inhibitor is continuously synthesizing in the, means this regulatory gene, it is continuously synthesizing. So now next is this promoter. This will do the synthesis of, first is lac-Z. Now lac-Z, it will produce a enzyme, that is beta-galactosidase. So this first gene will produce an enzyme beta-galactosidase. What this enzyme will do, it will break the lactose, the beta-1,4 bond which is present in the lactose, into glucose and galactose. Now this glucose and galactose, it can be used for the various metabolic product, like glucose will go into the glycolysis, and galactose, it is also converted into glucose and will be utilized for the different metabolic reactions. Now so lac-Z, it will produce beta-galactosidase. Now next, this lac-Y gene, it will produce a permease. This will produce an enzyme permease. Now this permease, these are the channel, these are the channels which are present on the plasma membrane, and this will allow the transport of lactose through the plasma membrane. Now next gene, this LAC-A, this structural gene, this will produce an enzyme that is trans-acetylase. This trans-acetylase enzyme, it will transfer the acetyl group from acetyl coenzyme A to the beta-galactosidase. So these are the three enzymes, or we can say the protein, which are produced or synthesized by this operon. So now we will see the different condition. When lac operon is off. So when this lac operon will be in off condition, when there is no presence of lactose. So lactose is absent. In absence of the lactose, what will happen? This lac operon will be in the off state. So what will be happening in that condition? In off condition, what will be happening? This LAC-I gene, it is continuously synthesizing, means this inhibitor, it is continuously synthesizing, and now this will go and bind to the promoter region of this structural gene. So what will happen? Now this promoter can't work, and the different enzyme, this can't be synthesized. Now later on, in the next condition, when the lac operon will be on, and when this lac operon will be on, when there is presence of lactose. So when in the medium lactose is present, what will happen? This the lactose in the medium, when lactose is present, now what will happen? Some lactose will be converted into an allolactose. Now allolactose, suppose this is a allolactose. So what this allolactose is going to do? This will now come and bind to this inhibitor. So to the inhibitor, now what will happen? This allolactose has bind. Now this inhibitor, it can't bind to the promoter region of the structural gene, and there will this is of these three enzymes or protein, that is beta-galactosidase, permease and trans-acetylase. So this is how this lac operon will be working. Now next, second, we are going to see the that is the repressible system. So what will be happening in the repressible system? So in case of the repressible system, suppose there is an enzyme which will be present in high amount or in high concentration. So their end product, it will be accumulated in the during the reaction, and that end product, when their concentration increases, it will be acting as a co-repressor. Now this co-repressor, when there is a regulatory gene, it will synthesize a aporepressor. Now this co-repressor and aporepressor, they will act, they will form a active repressor, and they will be not allowing the synthesis of the mRNA. So we will see that. So this is the, suppose the enzyme, when it is present in high concentration, it will produce a end product, and the end product is acting as a co-repressor. When now this co-repressor, it is or the end product, when it is accumulated in the reaction in a high amount, what will happen? The end product of that reaction, it is accumulated or it is very high in the reaction. Now this will be acting as a co-repressor. Now a regulatory gene, it will produce to the co-repressor, it will produce a allorepressor. Now this will be forming a repressor or an active repressor. Now this will inhibit the transcription or synthesis of the mRNA. Now this in case of the salmonella, there will be this repressor system will be acting. So in the salmonella, there is a synthesis of histidine. Now this histidine this is an aporepressor. Co-repressor plus aporepressor, it will form a repressor. Now in case of the salmonella, what will happen? There is a production of this amino acid histidine. When this amino acid, it is present in high concentration, now this histidine, it will start acting as a co-repressor. When the concentration increases, this histidine, it will be acting as a co-repressor. Now there will be regulatory gene which will be forming the aporepressor. Now this two co-repressor and aporepressor, they will be forming an active repressor which will now further inhibit the process of transcription or the synthesis of a mRNA. So these are the two type of the that is regulatory system. First is inducible repressor, and next is first is the inducible operon, and next is the repressible system. So this comes under the negative regulation of a gene, that in case of the prokaryote. Thank you.